So welcome everybody to the Master's Defense for Kiley Jensen. Kiley is finishing his master's degree in entomology, working on some strawberry pests, a very, very important problem in all of pest management, known as pesticide resistance. And it occurs in plant pathogens, and it occurs in also all the arthropod pests that we have. Kiley did his bachelor's degree here at Cal Poly, was a dual plant sciences and entomology or plant pathology and ag science major. So when he came in ag science, he realized that it wasn't quite the fit that he wanted, so he involved the plant sciences department and made it dual, and then from there is doing his master's, finishing his master's in entomology. And he started that. His committee is comprised of Dr. Sarah Zukoff, who started that program with Kiley. They're pretty close to the end. And then I've stepped in as the chair of that department. We have David Hedrick and Fatima, who's also on the committee. And so we're glad to see everybody here. Ganji so far. That's right. Welcome, Fatima. We have a few other people on the Zoom call. So give it over to Kiley for his defense, and then we'll do the questions and answers after that, and then the committee will meet after that for more questions and answers.
Sounds good. All right. Hello, everybody. I'm Kiley Jensen. I'm a graduate student at the Cal Poly Strawberry Center. And today I'm going to be talking about my personal research project, which is the arthropod resistant status of Tetranychus urticae, which is commonly referred to as the two-spotted spider mite, and Lygus hesperus, which we will call just Lygus in this presentation. And when I talk about arthropod resistant status, I mean the resistant status which is heritable in these mite and insect populations. Resistance develops in agricultural fields by selecting for pre-adapted insects and mites in agricultural systems with pesticide applications. These resistant individuals survive applications, repopulate, recolonize, and become the predominant lineage in an agricultural system. So just to give a little bit of background on strawberries, where we grow strawberries in California, we're primarily concerned with three growing districts. This is the Watsonville and Salinas area, the Santa Maria and Tacoma area, and as well as the Oxnard and Ventura area. Strawberries are a very valuable crop. They have a consistent annual value of around $2 billion just in California. And we farm only 40,000 acres, which accounts for 90% of the production in all of the United States. And again, this is primarily produced in these three growing districts. The total operating cost to produce and harvest strawberries in the Salinas and Watsonville area in the year 2021 was $89,000 per acre. And California strawberry growers are really concerned with the best way to protect this large monetary investment from arthropod pest damages. Now, just to give a little bit of background on the biology of the mite species and the insect species and how we manage them in our California production systems. So the first species we're talking about, Tetranychus urticae, commonly called the two-spotted spider mite. You can see it here in this top image on this slide. They have a lot of phenotypic plasticity, meaning that they have a wide range of coloration that they can express. They can be anywhere from a light yellow to a green to this dark crimson red that you see here on the slide. And they have a very wide host range. They're established in 100 different countries, and they have 1,000 different host plants. This is one of the most well-studied pest species in the world when it comes to arthropod resistance. That's because it established itself in multiple agricultural systems, and that includes things like corn, beans, cucurbits, hops, and tomatoes. Those are some of the other important agricultural commodities that this pest infests. And how they do damage to plants is they feed on the mesophyll plant tissue layer. They insert their mouthparts, which are called chelicerae, through epidermal pavement cells or through stomatal openings, and they suck out mesophyll cell contents. This eventually results in that mesophyll cell layer collapsing, which is expressed as stippling, scarring, and bronzing in the plant. This bottom image that you see here on the slide This is a severe mite infestation just in the Santa Maria growing region. This image was taken in an organic field under hoops. And you have a really severe mite infestation when you start to see webbing on plants. This is a way that mites use to transport themselves. It increases their mobility and it hinders predators from attacking large colonies. And plants that are not treated with any sort of treatment, whether that be biological or chemical application, can experience up to a 40% yield loss with an average reduction of 30% due to mite activity. This mite feeding eventually results in a decrease in photosynthesis rate for the plant. And this, again, ultimately results in reduced vigor, plant yield, and fruit size. Some of the ways that we have to manage the two-spotted spider mite in agricultural systems include dust suppression. This is typically just mounting a water tank of some sort on the back of a tractor or on a truck, and they literally just dump water on the drive rows of their farms. One of the ways that mites can disseminate in fields is on dust particles, and this hinders that capacity. And augmentated biological control is also very popular with the release of several predatory mite species. The first species that we'll talk about is Phytocilius persimilis. That is this mite in this top image on this slide, the orange mite, large-bodied mite. You see it feeding actively on a male two-spotted spider mite species. And there are some complications with this predator, but it is relatively effective at suppressing two-spotted spider mite populations in agricultural systems. Some of these issues include susceptibility to chemical applications. It's not uncommon for predators to be released and then their numbers reduced by pesticide applications. And this species, Phytocilius persimilis, again, this orange mite on the slide, is an incredibly selective predator. It will only feed on pest spider mites in the genus Tetranychus, but we happen to have a second pest spider mite species in California strawberries, and this is known as Tetranychus lewisi. And that's that mite that you see in the very bottom right slide that's being fed on by a different predatory mite species. But Tetranychus lewisi damages strawberry plants in the same exact way that Tetranychus urticae does, but Phytocilius persimilis will not actually feed on it. So it's not uncommon for Phytocilius persimilis to be introduced to an agricultural system, for them to control the two-spotted spider mite population, and for a secondary pest outbreak of the Lewis spider mite to occur in those systems. And to combat that, we do release other predators. The predator in that bottom right image that you can see actively feeding on the Lewis mite is Lygus hesperus, which is not incredibly commonly released in California systems. But Neosalius californicus, which is that predatory mite feeding on a two-spotted spider mite egg in that bottom left image, is commonly released. This predatory mite, Neosalius californicus, is considerably more hardy than Phytocilius persimilis. It survives better in our agricultural systems, in our climate here in California, but it will participate in intraguild predation. So in mixed systems of Phytocilius persimilis and Neosalius californicus, Californicus has been shown to readily displace Phytocilius persimilis. So all that just to say that biological control doesn't quite control two-spotted spider mite to acceptable levels at all times in the production year. Moving on to the next insect, Lygus hesperus. This is the official common name is the Western tarnished plant bug, but again, we do not call it this in our California systems. We call it just Lygus. And they feed directly on the achenes of strawberry fruit. Every single seed that you see there on that strawberry is actually a nutrient sink, and Lygus feeding on these sites destroys that site as a nutrient sink. So what commonly occurs is Lygus will feed on berries early on in the development cycle of the strawberry. They'll destroy these nutrient sinks, and that portion of the berry will never develop. That's what you can see here on the strawberry in this image. That portion of the berry, which is being folded in upon, has been fed upon by Lygus. And as a result, it creates this, what we call a cat-facing effect. And this is unsaleable or unmarketable to fresh markets in California. Lygus can cause up to $102 million in damages in a single year. And this is a very low estimate. but they can cause $11 million in damages in a single week during peak production season in the summer. Some of the ways that we have to manage Lygus hesperus in our fields include alfalfa trap crops. In mixed cropping systems of Lygus, or of strawberries and of alfalfa, alfalfa is a preferred host of Lygus. So Lygus will readily migrate to alfalfa plantings, but this can be seen as a net economic loss for California strawberry producers. Again, strawberries are a very valuable crop, and for every row of alfalfa that you're growing in your field, it can be seen as a net economic loss. Another common tactic for Lygus control is quite literally vacuuming them out of fields using tracted mounted vacuums. Triple barrel vacuums are mounted on the front of a tractor, and the insects are sucked up out of the field, and on the very top of these vacuums, there are baffles. Whenever the insects hit these baffles, they're typically killed. However, this practice is non-selective, so it is not uncommon for predatory species to also be affected by tracted mounted vacuuming. This includes things like big-eyed bugs and geochorus species, minute pirate bugs, in other words, as these are also both hemipterans in the same order that Lygus is. And spiders are also typically negatively affected, which are another primary predator of Lygus species. Alternate host elimination has been shown to be effective, but again, doesn't suppress Lygus populations enough in California strawberry fields. Lygus is also incredibly prolificous, and it can feed on over 100 different species of plants, many of which are California native weed species. So elimination of weeds in and outside of strawberry fields can reduce Lygus numbers. Right now, all of that leading up to those management tactics typically fail to suppress Lygus and spider mite species below economic injury thresholds. Strawberries are such a valuable crop that economic injury thresholds are relatively low compared to other agricultural systems. The damage threshold for Lygus, these damage thresholds are typically a pest population density in a field, and the damage threshold for Lygus is a single insect within 20 strawberry plants, and for mites, it is 15 mites per mid-tier leaflet during the harvest season of strawberries. Over 9.5 million pounds of pesticide were applied in 2018 just to California strawberry production fields. This figure also includes fungicide applications, but fungicides can also be detrimental to the community complexes of predators in California strawberry production fields. And resistance has been a concern for both of these pest species since the 60s. Lygus resistance to malathion has been prevalent since 1968. Malathion is a chemical class that we still use today to control Lygus in our production fields. And alternatives to miticides were considered for two-spotted spider mite as early as 1966. And this is when augmentative biological control using predatory mite species was first popularized. All right, now moving on to what I actually did in my experiments, my materials and methods. I conducted laboratory bioassays. What these bioassays were artificial arenas coated in insecticide or miticide, in which I would then infest the pest species in question into these arenas and assess mortality after a specific amount of time. In most cases, this was 48 hours, but there were some exceptions to this. One of the insecticides that I tested was an insect growth regulator, and that chemical had to be checked after 7 days to ensure that mites would attempt to molt, or insects would attempt to molt in that time. And there are some key differences between the assays that I conducted on the mite species and the insect species. Most of the miticides that are registered in California strawberry production fields work with contact activity, meaning that the mites simply have to touch the miticide for them to work in the intended way. And then a majority of the insecticides are systemics. These are commonly referred to as stomach poisons, and the insects actually have to ingest the chemical for it to work properly. And how I'm actually detecting resistance in this way is by comparing the response of resistant field populations, the mortality response of these resistant field populations, to the mortality response of susceptible populations of the insect and mite species. I received susceptible populations, so these populations haven't been sprayed with any insecticide or miticide for over 50 years. And I received these populations, the population of spider mites from Washington State University, from Doug Walsh up in Pullman, and then the Lygus colony I received from Colin Brent at the USDA Arid Research Center. How I chose my dosages and chemicals were a little bit different between the insect and mite assays. First, to choose the chemicals that I was actually assaying, I surveyed California pest control advisors. A pesticide application cannot be legally permitted in California agricultural production systems without a recommendation from a pest control advisor. So these are really authority figures on what is actually being applied to California strawberry production fields. And we asked them what they are typically prescribing, as well as what they think is effective. And then we chose 4 insecticides and 4 miticides to test from this list that we were given. And how I got my dosages is a little bit different between the insecticide work and the miticide work. For the miticide work, I used active ingredients. Active ingredients are the killing agent of pesticide formulations. A common pesticide that many people know of is Roundup. Well, Roundup is a formulation, and the active ingredient of Roundup is glyphosate. So if I was doing herbicide trials in the same way that I was doing my miticide trials, I would have used glyphosate and not the formulation of Roundup. And then when it comes to the insecticide work, I used actual just formulations, so registered products that California strawberry growers can purchase and apply to their fields. And this would again be equivalent to just the formulation of Roundup. Also, something to note with my miticide work, the dosages that I used, I got from a dose response curve of a variety of chemical concentrations of these active ingredient miticides. I exposed the susceptible population to 8 concentrations of 4 different miticides, and then we fit a dose response curve to this mortality response and found out the concentration, which theoretically kills 50% and 90% of that susceptible population with that chemical. We then took these LC50 and LC90 values and actually exposed the susceptible population again, since this is a theoretical value and we wanted to see the actual response of the population, as well as field populations of 2-spotted spider mite. And then I compared the mortality response in that susceptible group with resistant field populations. And if there was a significant difference in mortality between that susceptible group and field populations, we would attribute that difference in mortality to resistance. And then insecticides were much simpler. We just used that registered formulation at the maximum allowable rate. And just some notes about the location codes that you'll see in the rest of the presentation. I coded Santa Maria as SM/NEPO, Ventura as VEN, and Salinas as SAL. I collected 2-spotted spider mite populations from Santa Maria and Ventura, and I collected Lygus populations from Santa Maria and Salinas. So how I actually conducted these bioassays for the 2-spotted spider mite. I used the adult female life stage. This is the most susceptible the most resistant life stage to miticide applications. And these individuals are capable of producing progeny while unmated. So in isolated systems, a single adult female can give rise to an entire colony by themselves. So they're an important life stage to control for California strawberry producers. Again, the miticides work with contact activities, so I don't have to provide a treated food source in these trials. What I would do is dissolve the active ingredient of the miticides into acetone. I would place that acetone in these small vials that you see here in this bottom right image. We'd put those vials that have the acetone and active ingredient in them on a hot dog roller, and I would let the acetone evaporate off. This would typically take around 4 minutes. And then all that's left in the vial is pure active ingredient. I would then infest the 2-spotted spider mite using a paintbrush. I would cut a majority of the hairs off of the paintbrush and place the mites individually into these arenas and then seal them and assess mortality after 48 hours for all these chemicals that I did test. And we screened a total of 4 miticides. And again, the dosages that we used were determined from that dose response curve, which I will show shortly. All right, here's the chemicals that I tested. Again, the active ingredients are listed in the chemical column. So these are the names that I will be using when talking about these chemicals. And that is abamectin, bifenazate, spironolactone, and fenpyroxamate. As a general trend, the groups of these miticides are different. We wanted to test different chemical groups because as a guideline for California strawberry producers, they want to rotate these chemical groups when applying pesticides to their fields because this hinders the ability of two-spotted spider mite to build up resistance. This is because miticides listed in different groups actually kill the mite species in different ways, and they develop resistance typically differently between these different chemical groups. So it's possible that we could have seen trends where we would see exacerbated levels of resistance to the group 6 miticide, abamectin, but relatively low resistance cases in fenpyroxamate. And this would have allowed us to recommend California strawberry producers on a regional basis what miticides they should be looking to apply. Right, and again, here's that dose response curve with the susceptible colony. Every single one of those lines that you see are a different active ingredient on that graph. On the x-axis, we have the percent chemical concentration, and on the y-axis, we have the proportion of mortality. The steeper the line is, essentially the more toxic it is to the susceptible mite population. The black line is abamectin, the green line is bifenazate, the blue line is fenpyroxamate, and the yellow line is spiromesifit. That red line that you see at the top of the graph, that is the rate at which 90% of the susceptible population is actually killed. And where that red line intersects the dose response curve lines, that's where we get the theoretical percent concentration from that kills 90% of the susceptible population. And as a general trend, most of these miticides did reach that 90% mortality rate, but spiromesifin actually did not. So I cannot actually show you guys a 90% mortality response with that chemical. So I will just be showing you the LC50 value from that miticide. The symbols that you see associated with the lines on this graph are the actual data points that I got at a particular chemical concentration. And as you can see, none of the data points for spiromesifin were above that 90% concentration line. And here's the data from that dose response curve extrapolated and given a little bit more context. So again, in the left column, we have the chemicals that I tested, abamectin, bifenazate, spiromesifin, and fenpyroxamate, followed by the lethal concentration that killed 50% of that population and the lethal concentration that killed 90%. Again, the only exception where we didn't get an LC90 was spiromesifin. And to give you guys context of what we actually apply of these chemicals in California strawberry systems, we have the trade name of a particular active ingredient along with the parts per million of what we can apply to our agricultural systems with that miticide. So let's say the susceptible population of spider mites was infesting California strawberry production fields, and we applied abamectin at a rate of 14.6 parts per million. Well, the data from my dose response curve tells us that theoretically we could achieve over 90% mortality if we had a perfect pesticide application. And this is a general trend for most of the miticides that I tested. The LC90 of abamectin was half of what we were allowed to legally apply in strawberry fields. For bifenazate, much of the same, just about half of what we're allowed to apply in an ag system is what I got that killed 90% of the susceptible population. Spiromesifin, again, the exception. It was nearly 8 times higher. The rate was 8 times higher than what we're allowed to legally apply that killed 50% of that susceptible population. And fenpyroxamate, it was nearly 20 times lower, that LC9 felt there. All right, now the rest of the graphs for the rest of the presentation will look just like this. This first chemical is spiromesifin, and this is the chemical that we did not get an LC90 for. So I'm just showing you the LC50 results here. On the x-axis, we have population, and on the y-axis, we have proportion of dead two-spotted spider mite. The most important value on this graph is the response by the susceptible population. That number will always be left justified on these graphs. So again, we got that theoretical LC50 value from the dose response curve. Then we actually exposed the susceptible population to this theoretical value, and this is that actual response. So for the susceptible population, this again is the concentration, which is supposed to kill 50% of that susceptible population, and that's just about the response that we got with the susceptible population. And if there were resistance, so if there were a significant difference between the susceptible population and field populations on this graph, there would be an asterisk above the error bar of that field. So for this chemical at this concentration, we were not capable of detecting resistance in any field populations that we tested. And these field populations that we tested, 5 were sourced from Santa Maria and 5 were sourced from Ventura. All right, and this is the next chemical, abamectin. I'll be showing you guys the LC90 values for the miticides from here on out. Again, the actual response of the susceptible population to this chemical at the LC90 concentration was just about what was theoretically expected from that dose response curve. And we were actually able to detect resistance in several field populations with this methodology. We detected resistance in the populations 1SM, 3SM, and 5SM, as well as 1-ven, 2-ven, 3-ven, and 5-ven.
Oh, so they're just susceptible.
Mhm.
It's right at that.
Always left justified, yeah.
And then everybody else is
Being compared to that susceptible.
lower, but only significantly lower.
Yeah.
So what's with 2S not being significantly? Oh, I see.
It overlaps.
Okay, gotcha.
Yeah.
Okay, thank you.
All right, this next chemical, this is bifenazate at the LC90 concentration. The theoretical response by the susceptible population was higher than what was expected. Over 95% mortality was incurred. And we were incapable of detecting resistance in any of the field populations from Santa Maria. And we did detect resistance in 3 populations sourced from Ventura. This next chemical, fenpyroxamate, this is the last miticide I'll be showing you guys. The theoretical response was just about correct again with the susceptible population. 90% mortality was incurred. And there was resistance detected in 3 populations from Santa Maria and 4 populations from Ventura. Right, now moving on to Lygus. I had to group Lygus into 3 different life stages. One of the chemicals that I tested, one of the insecticides, has no activity on the adult life stage of Lygus. So I created these groups and exposed them to this chemical. I only exposed nymphal life stages to this single chemical, which was not active on adults. So I grouped instars 1 through 3 in a small, what I called a small nymph group, and instars 4 and 5 into a large nymph group. Again, most of these chemicals, most of these insecticides work with systemic activity. So I did have to provide a food source in these trials. And that's what you see here in this bottom image. Those are what my bioassay arenas looked like. And in the same way that I detected resistance with the mite work, I am comparing the mortality response in a susceptible population to field populations of Lygus sourced from California strawberries. We screened 5 insecticides, and one of these was omitted because there was very low mortality response in the susceptible population. And in those cases, we were incapable of discerning whether or not there was resistance, since we are comparing mortality to the response in the susceptible group. And one of the chemicals that we did test for 2 years, I'll only be showing you guys data from the year 2022. The mortality response in the susceptible group for one of the chemicals that I did test and will be showing you guys was incredibly low. And as a result, we created a modified assay. And in this modified assay, we infested a food source as well as the insects into an arena with openings at the top and bottom. And we submerged this container entirely in insecticide, let it dry, and then assess mortality response after 48 hours. In this image in the bottom right is that modified array. Here are the insecticides that I tested. Again, I'm using the registered field formulations of these insecticides, not the active ingredients. So I'll refer to them as their trade names. That is Ryman, Savanto, Brigade, and Actara. We don't have that same group trend that we had with the miticide work. There are 2 group 4 insecticides here on this that we tested. But there's actually no concerns of cross-resistance between these 2 chemicals because they're in different subclasses and the insects develop resistance to them differently. The activity of the majority of these are systemics, and most of the products are selective towards piercing, sucking insects. You'll notice right away that there are red asterisks on this side instead of black. That's because we couldn't make sound statistical conclusions with some of these field populations. That is because the response of the susceptible population was 100% mortality. And in these cases, we couldn't confidently calculate standard error. And as a result, we have to just assume that there is resistance in field populations rather than making sound statistical conclusions. So when there's a red asterisk above a field, we are just assuming resistance in that field population since mortality response by that field population was not also 100%. So we are assuming resistance in all the field populations that we tested. And again, this is my data from 2022, and that was 3 populations from Salinas and 5 populations from the Santa Maria region. This next chemical, sorry, I forgot to explain, but that first chemical was the insect growth regulator, tested against the small nymph group. And now this is that same chemical tested against the large nymph group, so instars 4 and 5. The mortality response by the susceptible population was very high, but not quite 100%. So we are able to make statistical conclusions in this case. And in this way, we detected resistance to this chemical at these life stages in the populations 1 sal, 2 sal, 3 NEPO, 7 SM, and 8 SM. So 2 populations, 2 out of 3 populations from Salinas, and 3 out of 5 from the Santa Maria region. This next chemical, there's only a few fields here on the slide because this is the chemical that created that modified assay halfway through the data gathering year. Why we created that assay, again, was because the susceptible response in this group was relatively low. Just about 30% mortality was incurred by the susceptible population at the maximum allowable field rate with this chemical. This is a butanolide, by the way. And this is that residual assay that we tested. But we are assuming resistance in 2 of the field populations that we tested since there was 0 mortality response in these groups. This is that same chemical again, but this is with that modified assay where we would submerge the insects entirely in the insecticide in that modified arena container. But we again ran into the issue where 100% of the susceptible population died, and as a result, we can't make sound statistical conclusions. So we are simply assuming resistance in all the field populations that we tested. This next chemical, brigade, bifenthrin, this is a pyrethroid. The mortality response to the susceptible population was relatively high, just about 80%. And we detected resistance or assumed resistance in all the field populations that we tested. Bifenthrin is a pyrethroid again. This shares a target site with the chemical DDT, which was used to control Lygus species in California as early as the 1940s. So it's possible that Lygus populations have been building resistance to this chemical class since that time. And this final chemical I'll be showing you guys, Actara thiamethixim. This is a neonicotinoid. The mortality response was just above 75% in the susceptible group, and we are detecting or assuming resistance again in all the field populations that we tested. All right, moving on to the discussion for the 2-spotted spider mite. Resistance was detected more often in the Ventura region than in the Santa Maria region. This could be explained by the fact that Ventura has a staggered production system for strawberries. They'll plant nearly half of their acres in the fall and half in the summer. So it's possible that these 2-spotted spider mite species are readily migrating between these 2 production systems and being managed on a continual annual basis, which exacerbates resistance. Resistance detection was difficult since only mortality was measured in my assays. There are many secondary effects of chemical applications which were not measured, and that includes things like feeding cessation, decreased mobility, and reduced egg line capacity. But again, I only assessed mortality in my assays. And spiromesipin, that chemical that we didn't get an LC90 for, I just showed you the LC50 products. This chemical has vastly well-documented secondary effects that are not associated with just mortality. And it's possible that mortality would have been occurred if I had waited to do the check longer than 48 hours. LC50 results were omitted from this presentation for the miticide work, and that's because they're somewhat contradictory to what we found in the LC90 group. The LC50 response, it was much more difficult to detect resistance Because control mortality was relatively high, nearly 20% control mortality was average in my experiments. And there was relatively low statistical power. And as a result, it was hard to make statistical conclusions between what could have been attributed to control mortality and the mortality that was expected with the treatments. Since there was a relatively low number of replication, since I was testing so many miticides in concentrations. And then for Lygus, statistically sound results were hard to produce because of that 100% and 0% mortality response from the susceptible populations and field populations. The efficacy of Ryman was the most promising for the control of Lygus, but this is the chemical that we tested against the nymphal life stages of Lygus, so the small nymph and large nymph group. And as a general rule of thumb, insecticides are typically more effective against nymphal life stages. So for future work, this has been recommended by extensionists in the past, but California strawberry growers could expose their mite populations to concentrations similar to what I did with my susceptible population, and then compare the mortality response. This isn't typically done, but it could save growers a lot of money if they decided to test the efficacy of their insecticides and miticides before they actually make an application. And if these assays were to be rerun, there should be increased replication and LC99 concentrations instead of LC50 concentrations should be used. And then for Lygus, efficacy and resistance testing against the nymphal life stages would be beneficial. The University of California recommends that insecticide applications be targeted at the nymphal life stages in the first place. All right, and this is my committee. Dr. Zukoff was my chair for the majority of the project. This was later replaced by Dr. Holmes. And then my committee members include Dr. Hedrick and Dr. Conjasfar. And Miriam Mendez from the California Strawberry Commission helped me source a majority of my field populations. Every field population from the Santa Maria region was provided by her. All right, questions?
We have questions from either folks on the Zoom or in the room. We want to start.
We can hear you guys if you talk on Zoom. So
I'm curious about melathion. You mentioned that it started showing resistance, or Lygus started showing resistance in the 60s, but it's still being used today. Why is that? I'm just curious, like if you have an answer.
It's an organophosphate, which is mediated by what is it called? Metabolically, typically. Majority of the insecticides that we use in our California strawberry fields are mediated metabolically. So bifenthrin, that pyrethroid that I showed you, as well as the neonicotinoid thiamethixam, Actara, that's what I called it on that slide. Those are also both mediated metabolically for resistance. And while they are in different classes, that could explain why there's like exacerbated resistance levels to these compounds, since the insects are developing resistance to them in the same way. Why it's still registered, I would assume that has more to do with safety and the low REI of the chemical. More
Okay.
than.
So first, I don't know why I haven't asked this question before, but when you were showing those pictures early on in your presentation, you can see the size of the prey and the size of the predator.
Mhm.
And the prey is almost as big as the predator.
Mhm.
So how is this predator, how many individual prey are they consuming over a period of time? How voracious is this appetite? Because it looks like the first one you showed, I think it was Persimilis versus two spot, and two spot looked like it was about half the size.
Yeah, that was a male two spot, which are smaller than the adult females.
Wow. So how many female or male two spot can Persimilis consume?
I don't know when it comes to the various life stages. Typically, I've seen them more selective towards the egg life stage, and I've seen them consume 15 eggs in a day.
In a day?
Yeah.
That's incredible, because that egg looks like it's about a fifth of the size of the predator. So that would be like us consuming,
by body weight, yeah.
you know, like our own body weight every day, right?
Yeah.
So they're burning a lot of calories.
I have a question about the prevalence of Lewis mite and TSSM
Mhm.
and during your visits. Were you able to know who's more prevalent?
No, not really. We really didn't see a lot of Lewis except for flare-ups in individual fields, especially in Santa Maria. Supposedly it's more prevalent in Ventura, but Carolina actually provided my Ventura populations for me, so I didn't actively go to Ventura and scout fields myself. But it was pretty rare that we would be told about a Lewis infestation. Early on when I first started my project, I did plan on doing these assays against Lewis as well. But it was difficult to sustain the populations and to source them.
Kylie, I have a question. Can you hear me?
Yeah.
Okay. So in general, you see more resistant in Ventura area. Do you have any idea what's going on in there? Do they have different insecticide application, like more free? Do they apply insecticide more frequently, or yeah?
Yeah, so I mentioned that a little bit with the staggered production system. So they plant just pretty much half their acreage in the fall and then half in the summer. But as a general trend, we have seen less Lygus in the Ventura region. That could be due to temperature. It's much hotter down in the Ventura region than, say, the Salinas and Santa Maria regions. So it's possible that they're relying more on specifically miticides to control the two-spotted spider mite populations, and they're not applying a lot of insecticides, which could, rotate the mode of action and decrease that incidence of resistance to just miticide applications.
Thoughts on why that the miticide spider,
Spironectin.
that one.
Yeah.
Why it was not meeting the LC90 and why it was already seeming like it maybe not resistant, it just wasn't working.
Yeah.
Why was that?
So on the label Oberon, it does suggest that you do target nymphal life stages and egg stages.
That was just you were just
Yeah, and I was just using adults. And it's really reproductive secondary effects that are well documented with that chemical class. Yeah, not just mortality.
That last rad.
Mhm.
Just curious why you have both aspects in both colors.
Yeah, so in the cases where there was 0% mortality response, we couldn't make that sound statistical conclusion because we couldn't calculate standard error. So it went both ways, when there was 100% or when there was 0%. We couldn't make that distinction. Yeah. So we're just assuming resistance whenever there's red.
So what are you going to tell the growers? Let's say, what are you going to tell the growers in Santa Maria versus what are you going to tell the growers in Ventura? Or two-spotted spider mite best management practices based on the information you have here and Lygus.
I would have to go chemical by chemical. I really didn't look at it on a growing region only basis. I was mostly comparing between growing regions. So I would have to really dive into the results in those particular regions and then compare it to what has been recorded historically for those regions too, before I made, a recommendation, I would say.
Okay, so that's how you play it safe.
Yeah.
So,
No?
so you're just having coffee with the grower
Right.
from Santa Maria, and he says, okay, you've been doing this resistant stuff. What should I spray?
Uh-huh.
And let's say it's Santa Maria and it's two-spotted spider mite. what are you gonna tell that grower?
I would show them grass chemical by chemical first off. Let's see if I can pull this up. But I would really recommend immediately for them to do their own bioassays to determine if they're going to make a miticide application. But the miticides in general had a much higher efficacy than the insecticide work. So I would personally, if I were a grower, I would be less concerned with miticide resistance than I was, say, Lygus resistance. So I would say follow your rotational typical
So
actions.
you wouldn't drop any of them out, you would say, keep on the rotation.
I would emphasize don't target spiromesipin applications at adults.
So then you
Especially-
gotta know what population structures are.
Yeah.
So you're gonna tell him or her that, they need to pay somebody to be monitoring so that they have better, targeted times.
If two spot was becoming that big of an issue and they could justify that, I would say definitely.
You say you would have them do their own bioassays. You talking about a bioassay like the one you did? Or are you talking about something a little different that a grower could actually do?
There's faster and dirtier ways to do what I did. I've heard of Frank Zalum's told us that he used sandwich baggies. He would put pesticide in sandwich baggies and then put the bugs in.
Okay.
So something like that. Whatever container they have lying around, that preferably it wasn't a food container of some
So it's
sort.
sandwich bag that is coated with your
Yeah.
insecticide, or
Yeah, or a food source
or
that's
food
coated.
source.
Yeah, and then just place inside. Yeah.
Okay. Is anybody doing that?
It's been recommended for years. I don't know if
And
anyone
is
that actually
anybody
does
doing
it. I don't know.
I've never seen any PCAs
Yeah.
with resistance assays in there. I think that's pretty cool though, to be able to do that. You get an answer in 48 hours, right?
Yeah.
You didn't have it bacon on your dashboard.
Yeah.
Check about the chemicals that were resistant with the PCAs and add if they, or check their pesticide neutral codes. Actually, those things.
With what I was- No, we didn't do that. So for the mite work, that wasn't really a concern, simply because we took the mites from the field and then reared them ourselves in tents. So it's possible that previous applications would influence, of course, resistance that we saw. But for the Lygus work, that could have been more of a concern because we took insects directly from the field and placed them into our bioassays. So it's possible they could have been exposed to a chemical as early as last week, which could be a confounding factor of why we're seeing mortality.
To follow up on that question, did you collect your Lygus all from conventional? I might have missed that. All from conventional fields, or did you try to see if there was a difference between conventional and organic fields? I know you said you had different, like for right now on the screen, you have 1 ven, 2 ven, 3 ven. So I don't know if they're all conventional or if some of them are organic.
About half of the mite fields were organic. I don't know exactly what those are offhand, but all of the Lygus that I sampled were from conventional systems.
Okay, nice.
Question about Lygus feeding.
Mm.
So they feed on the achenes. Have you ever seen them feeding on an achene? And can you tell an achene that has been fed on versus one that hasn't?
Not immediately. To tell that the stylet of Lygus is incredibly small. There's images of it with scanning electron microscopes. One researcher, I think they used liquid nitrogen. They took a Lygus that was feeding on a berry or something and then dipped it into that liquid nitrogen and then used that scanning electron microscope to see it actively feeding. But the feeding incision is microscopic.
So that stylet is microscopic diameter?
Yeah.
Yeah, because I don't see it in the achenes, but I look at a lot of fruit and a lot of achenes. Have you ever seen them in the act?
I've seen them feeding in the assay arenas, yeah.
Okay.
But they'll also feed on not just achenes, they'll feed on receptacles surrounding achenes. That's a preferred feeding site, but they'll feed on normal plant tissue as well.
Does the akene, after feeding, like day or 2 or 3 or 4 later, start discoloring?
I'm not sure, honestly. What I mostly know is that we can tell it apart from pollination damage because the size of the akene remains consistent with the rest of the berry, whereas achenes that never developed in the first place are tiny, are they not undeveloped? Yeah. I'm not sure about coloration.
So this feeding causes the cat facing of the fruit. What else will cause cat facing?
Pollination,
So what causes pollination damage?
just a lack of that individual ovary being fertilized.
And what would cause that?
Well,
Can it
typically,
be confounded? And can the things that cause cat facing that aren't Lygus be confused with the cat facing that Lygus causes?
you can tell the difference, but California strawberry producers cannot always. So Lygus can be used as a scapegoat for what is pollination damage.
So it's pretty common to have cat facing even if there's
No Lygus.
no Lygus. And what might be under what conditions might you see a lot of cat facing?
I would assume a lack of wind when the
Lack of wind?
whenever receptacles are responsive.
So in other words, pollen isn't being transferred.
Yeah.
Okay. What else? Anything else that might?
Those are the two primary ones. Thrips have been blamed on cat facing, but they don't actually cause cat facing. Just bronzing.
What else might cause lack of pollination? Besides no transfer of the pollen.
Maybe the ovary aborting, fungus infection, too much heat, something like that.
Fungal infection, high temperatures,
Yeah.
and are those common?
Maybe this year.
They're common every year at certain times. At certain times,
Yeah.
right? And this is a point of contention
Right.
out there that people say, "Oh no, that's Lygus." Well, it's been hot.
Uh-huh.
Oh, all these other things. So, and I've always wondered if it's a different type of cat facing, because some kinds of pollination problems result in a kind of small button shaped fruit where the tip seems to be the thing that the pollination at the tip was aborted, so it becomes this kind of almost like a flattened pumpkin squashes. And you see that I see it, and this is just anecdotal, but hot, when it gets really hot, that's the kind of malformation that I see. And I'm wondering if that is more of a result of heat damage versus a real contorted looking fruit, true cat facing, let's say, because just the shorter, squattier berry doesn't necessarily look and sound like cat facing, it looks like cat facing. Anyway, there's all kinds of interesting things there, and I think it still eludes us a little bit. You know, it's not all nailed down, wrapped up. So even I've taken lots of pictures of cat-faced fruit and sat down with an entomologist who really knows, and I'll say, is this one Lygus or is this one something else? And neither, it's not that easy to figure it out. But what you're saying is that when the achenes are the same size, but you still have a cat-faced fruit, that's the real marker.
We attribute that to Lygus.
Yeah. And I think that it's easier said than.
Right. Some of the unpollinated ones are obvious though.
Yeah.
I have a question about your comment on how Ventura population of Lygus is different from Santa Maria. You commented that the Lygus population might be migrating from from fall to summer production.
Right.
But my understanding is that Santa Maria also has a summer production.
Right.
Why are we seeing that in the Santa Maria region?
I would assume that's based on climate, so just temperature and humidity and those effects on the populations. Santa Maria also has a lot of production of side strawberries, which can also be hosts of Lygus, things like celery. So it's possible. So the Ventura's auxiliary region is typically more concerned with mite activity than Lygus actually. Lygus, the most damage occurs from Lygus in the Salinas area, but that's because they have the longest production season. And then Santa Maria is planting more and more acreage every year, which could possibly upset that trend of where that Lygus damage is actually occurring. So I would say it's mostly host availability and temperature differences between regions.
You have a couple of pictures of bug vacuums I want you to go back to. Okay, so there's that picture. And then I think if you go back one more, there's another one, yeah. So there's really different. There are different vacuums and there's differences in the positioning of it. What's going on here? The vacuum is really high.
Mm-hmm. So he's just turning around.
Okay, so
Yeah,
he's raised
exiting
up
his
so he can
yeah.
turn around.
Yeah.
Okay,
Yeah.
I want to make sure
But typically they're this close to the bed during the actual process.
down
Yeah.
low,
Yeah.
so they can
Yeah.
get suction.
Yeah.
Otherwise, no suction.
Yeah.
No light. Right.
Mm.
And what else is different about you mentioned, you said, I thought I heard you say triple barrel.
Yeah. So just
what is the triple?
a vacuum that spans 3 beds.
Okay.
Yeah.
Because they started using the words double barrel or 2 barrels above one barrel.
Right.
Yeah, so when you said 3, I was like,
Right.
3 barrels. Okay,
Yeah.
this is what you're calling a triple barrel.
Yeah.
I would, to avoid the confusion of
Right.
terminology, I would call this a 3-bed
Yeah. Right.
vacuum. That way we don't
Right.
get the double barrel, triple barrel.
Don't upset the automation guys. Yeah.
And so I see the baffles there right up against the top of the vacuum. And then if you go back to the other one, you can see that the baffles are way above, they're raised above it. Do you know why that is?
Not
Do
really.
you know how they optimize vacuum for Lygus control?
No, I only ever read 2 studies, and those are just field efficacy studies. One was by Zalen, and then one was by I think Pickle, one of the old students.
Just wondered how familiar you were with the other methods that have been used to control Lygus.
Right.
When it comes to mites, you mentioned that the Persimolis is used. Sometimes doesn't always control 2-spot spider mite, but usually does. Can you talk a little bit about how effective is this method of control industry-wide? Is it doing the job?
They're very effective, but they're very fragile species. So if environmental conditions aren't perfect, conducive for their success, they'll die out very quickly. They're very susceptible to insecticide applications and residual applications. So if you release them too early, it's possible they'll just all die off. And they readily migrate whenever there's not enough 2-spot around. So that's why they'll just leave production systems quickly if there's not enough prey for them. And the other predators can also influence their presence. So it's a complicated system.
So does it happen? How frequently does it happen that you don't get the control you want from this?
I'm really not sure. Like, there's such a wide range of release rates that could influence something like that. I've seen as low as 30,000 per acre recommended, and as high as 100,000. Some people spot treat, some people do broadcast applications. There's so many factors that
But you've been working in the lab where they've
yeah.
been investigating some of the reasons why this doesn't work.
Mhm.
I'm just curious, do you have a sense, as you've worked in this lab and in this industry, do you have a sense of how effective this is? Is it pretty much 90% effective and you just have an occasional mishap? Or is this all over the place? Or is this like rarely working? Or
They're incredibly effective if you can keep them alive. I think that's really what it comes down to. We've done assays in the lab using these guys to examine, their fecundity, how many eggs they can lay, how much they'll eat. We get super high mortality response, even in controlled conditions, which is optimized for them. Just a little bit too much moisture and they'll die.
Do you see people releasing both Californicus and Persimois at the same time, or as a follow-up?
Californicus will readily migrate into strawberry fields. So even if you don't release them together, sometimes Californicus can move in. Yeah.
Do they play well here?
No. Californicus will feed on Persimois.
They will.
Yeah.
Is Ryan safe on this is the privacy and
I'm really not sure. That one is the growth regulator, so it might be safe through the adult stage, but I doubt it for any other stage. I don't know how selective that chemical is. I know there's like 5 orders that are listed, but I'm not sure about the predators.
well, I think there's a few other things that influence the efficacy of this management technique. Besides, when you say if they survive, what about them being repelled by something?
Yeah, residuals can do that.
Yeah, it's an environment they did not enjoy.
Yeah. Even toothbot in bioassay experiments, they've been known to abandon leaves that are treated.
I think Sarah was showing that maybe some of the fungicides that are used are repelling the predatory mites. And perhaps that's repelling them to a degree that makes them less effective in the significant.
Yeah.
I don't know to what degree we would want to blame that, but certainly that's something to consider.
Yeah.
Makes some sense.
I've pointed people towards Copert as on their website, where you can compare chemicals and the susceptibility of their predators to those chemicals. Yeah, Shishika's is the one that showed me that actually, but I'm not sure how well vetted that source is.
Yeah. A lot of work needs to be done.
Right.
Yeah, some of it's good, it's just anecdotal.
All right, I think we've had enough questions for Kiley for this section of the thesis defense. I want to thank everybody for coming, and for those on Zoom, appreciate you joining us for this for Kiley's defense. We're going to close this portion of the defense and thank Kiley once again for an outstanding presentation. And then we'll take a break and go on to some committee. So I think you have another link for Fatima to join that section. Okay, so we'll close this. Thanks, everybody.
Cool.